So we're going to continue through the lecture series on evidence of evolution. This will be part 2 of that lecture series. So we had just, in the previous lecture, finished up talking about comparative anatomy and identifying or discussing these homologous structures or analogous structures in adult forms of different species. Embryology is another area of consideration when considering the evidence of evolution. That's because there are some anatomical homologies that are not visible in adult organisms. Rather, they can only be seen during the embryonic stages of development. Embryos, again, they're the fertilized egg that will or is in the process of growing into a new individual. So embryology kind of observes or studies the early stages of development of organisms, and many of these embryos in different species resemble each other. Closely related organisms will actually go through similar developmental stages early in development, and they share many similar features. So, for instance, all vertebrate embryos have gill slits or pharyngeal pouches, which are these little kind of knobby looking things here, and they all have tails. They also all pass through different stages, which represent larval fish. With humans, the tails will kind of be absorbed, and that embryonic tail will become the tailbone, and those gill slits will turn into jaw and inner ear. So again, they're not visible during adult form. They're only visible during the embryonic stage. And these homologous embryonic structures, again, reflect that the developmental programs of vertebrates are variations on a similar plan that existed in their last common ancestor. So we are considered part of, or vertebrates, I should say, are part of Deuterostomia. Deuterostomia is a subtaxon of the group Bilateria. Bilateria is known for their bilateral symmetry. Now, vertebrates are part of chordates. So here's your early deuterostome, and then you have the division of or separation of your echinoderms from your chordates. Echinoderms lost or had that secondary loss of the gill slits. Chordates retained those gill slits. With your vertebrates, they are separate from your chordates in that, again, they don't retain the gill slits in their adult form. Gill slits are also these, it's just another term where it's in reference to the pharyngeal pouches, or pharyngeal, however you prefer to pronounce it. I always say in biology and various sciences, there's like 100 different ways to pronounce the same word. Anyway, so again, these gill slits, they're present in vertebrates only during the embryonic stage of development, and then slowly as you progress through those different, FIGARO, is that really necessary? Stop it. I'm sorry. As they go through embryonic development, the gill slits are kind of will develop into different adult form structures. So it's not that we really lose these gill slits, it's just that they become different structures. So you're probably wondering, okay, well, what do these gill slits or your pharyngeal pouches differentiate into? Well, in human embryos, there's actually 6 pharyngeal pouches. Only 4 can actually be observed on the surface. So you have the first pharyngeal pouch, or the mandibular arch. That's this one here. Your second pharyngeal arch, or the hyoid arch. Then there's the third, and then the fourth is kind of tucked in between. Here's the fourth right here. There's also fifth and sixth. Again, they're not observable at the surface, so you don't usually see them on these figures. But each of these pharyngeal pouches, again, develop into or give rise to an adult form structure. With the mandibular arch, so that's your first pharyngeal arch cartilage, that divides into the maxillary process and mandibular process. Now, your mandibular process differentiates into the malleus, incus, the sphenomandibular ligament, and then the mandible itself, which is your lower jaw. Maxillary process yields or gives rise to the maxilla, which is upper jaw. It'll give rise to the zygomatic bone, which is your cheekbone, and also the squamous part of the temporal bone will lie right around here, right next to and kind of anterior of the ear. The external auditory media, so your external ear canal, that's a derivative of the first pharyngeal fold. FIGARO. Really? Stop killing the squeaky dinosaur. I'm going to take it away. I'm going to take it away. The pharyngeal pouch, so the pharyngeal pouch of that first pharyngeal cartilage gives rise to the middle ear auditory. FIGARO. Hey. Okay, let me take the squeaky toy away. Go sit in the corner and think about what you did. Yes, you've lost your squeaky toy privileges for now. Okay, I'm sorry about that. Let's see, where were we? So the second pharyngeal arch, which is this guy here, also known as the hyoid arch. So as you can tell from the name, it's going to give rise to part of the hyoid bone. It will actually give rise to the lesser commu of the hyoid bone. It'll also form your stapes. Again, that's the kind of the last middle ear structure. The styloid process and the stylohyoid ligament. The third arch, so that's this one here, that's going to form the lower body or remaining portion of the hyoid bone. The fourth arch will give rise to the cartilages of your larynx and the cricothyroid cartilage. So here's your thyroid cartilage, here's the cricoid cartilage. This will also, your fourth will also fuse with the sixth arch to give rise to these structures. The fifth arch is rudimentary and will disappear later, and there's no derivatives of that arch. So again, these all form, or your gill arches, your gill slits will kind of give rise to these adult form structures, except when they don't, right? So there are cases where you have some improper fusion, or there is some irregularity in the embryonic developmental process, or during human embryogenesis, something goes wrong, doesn't end quite as it should. And this is where we see vestigial gills. A great example are branchial cleft cysts. So I actually had the pleasure of working with a patient who had a branchial cleft cyst since birth, and never gave her any issues. It would just kind of leak this translucent fluids. It was more of a nuisance than anything. but that was a vestigial gill slit. So branchial cleft cysts or fistulae, they often have tracts that extend from a site of embryologic origin. They're these congenital kind of epithelial defects, which arise on the lateral aspect of the neck when the second, typically it's the second branchial cleft, fails to obliterate during embryonic development. So phylogenetically, that entire branchial apparatus, meaning the opening and then that embryonic tract, they're related to gill slits. Now, the patient I worked with had the third brachial fistula. So hers was more of a hole that was situated kind of more towards the superior aspect of her clavicle. But there are some individuals who can have this occur with the first branchial fistula. And again, it's just the failure for those to fuse or obliterate during embryogenesis. Another example of vestigial gills are what are known as preauricular sinuses or a preauricular pit. This is a small hole that's present in front of the ear, and it occurs as a result of improper fusion of the first pharyngeal arch during the 6th week of gestation, if I'm correct. And that's when the ear is actually developing. Now, this is very rare. There's only about 1 to 10% of the human population actually has this occurring. So if you have it, you're one of the 1 to 10% of people, so you're really cool. Yeah, so those are vestigial gills. Now, one of the most celebrated cases of embryonic homology is what I call the fish gill, cartilage, reptilian jaw, and mammalian middle ear. A lot of people don't understand or don't recognize that our middle ear structures are homologous to early fish gill cartilages. So early fish, such as those that were ancestors of your modern lampreys or hagfish, they didn't have a jaw, right? They were anathostomes, meaning without a jaw. What they actually did was kind of modified food filters to form gills, and those gills acted to filter food particles from the water. Now the gills were composed, there were gill slits, right, which were kind of these little holes, which is where the particulates would actually move through, and then the gill slits themselves were supported by a series of gill arches, which were formed from cartilage or bone. And the gill arches also acted kind of in this anathostomous condition, not only to support the gill slits, but also the cranium or the skull of your early fish. Now, the actual evolution of jaws, that arose when these gill arches of the anathostomes became modified to form an actual jaw. And so the kind of mechanism that's proposed for how this occurred, it's thought that the first kind of gill arch or the mandibular arch became fused to the cranium. So it fused to the underside of the skull. And over time, it kind of enlarged with cartilage and bone and was squeezed and then hinged at the middle to form the basis of the upper and lower jaw. So this enlarged mandibular arch or your enlarged first arch again became the basis of your early jaw in the jawed fish. That second gill arch moved forward, and the second arch is also known as the hyoid arch. So that hyoid arch moved forward and extended from the quadrate bone, which was kind of situated at the rear of the cranium, and formed or molded around the angle of the jaw and acted to support that early jaw. In your bony fish, this hyoid arch later kind of specialized into the hyomandibular bone, which served to brace the quadrate bone. Now, there's ample evidence of this, and ample evidence between the notion that the vertebrate jaw is, in fact, homologous to the gill arches of jawless fishes. First line of evidence is that both of these sets of bones are made from neural crest cells. Most other bones come from mesodermal tissue. Second, both of these structures form from the upper and lower bars that bend forward and are hinged at the middle. So if you look back on this slide, you look at kind of the structure of these early gill arches, and you have like upper portion, bottom portion, and they're bent in structure. They're kind of bent right at the middle, which gives kind of support to this notion that your first arch, or that mandibular arch, over time was squeezed and then hinged at the middle, and that's what allowed for the upper and lower portions, and that kind of V-shaped or hinged structure. And the last is that the jaw muscular actually seems to be homologous to the original gill support muscular of the early jawless fish. So again, these are just kind of the major lines of evidence for the notion that our jaws and kind of all vertebrate jaws arose from the gill arches of jawless fish when they were modified to give early, or the first jaws in jawed vertebrates, which were first being those placoderms, which are these weird prehistoric fish that kind of had these big armor plates. It looked like their head was this giant shield, or kind of like the plated armor that you see knights wearing. Very weird looking. So not only is the vertebrate jaw homologous to gill arches of jawless fish, but hearing structures are as well. This is usually where people are like, you've got to be kidding me. And no, I'm not. So as vertebrates came up onto land, they had this new conundrum they were facing, right? This new problem was how to hear in a medium as thin as air. What to do? That's where things get interesting. So remember that quadrate bone I had just mentioned, the one that was right at the rear of the cranium? Well, as terrestrial vertebrates moved onto land and they began to alter their locomotion, their posture, that quadrate bone actually fused with the skull. And the purpose of that was to give a stronger bite. That's what it allowed for. Because the bone was now fused with the skull, it no longer needed a brace. So the hyomandibular bone, remember that was the brace for that early quadrate bone, lost its function for supporting the quadrate bone, which is very sad. However, the hyomandibular bone just so happens to be situated very close to the otic capsule. And it turns out that that bony material was excellent for transmitting sound. So while it lost its original function as a brace for the quadrate bone, the hyomandibular bone of your first tetrapods actually gained or acquired a new function as a sound transducer, transmitting vibrations into the inner ear. And it seems that that hyomandibular bone actually became, over time, specialized to form the stapes or columna of the reptilian ear, right? Reptiles have a single bone, which is the stapes, and that's used to transmit vibrations into the inner ear. Their jaw actually articulates as well with the stapes, and that helps to receive substrate vibrations. So that's just another mechanism of sound transducing. Now, one group of reptiles, known as the synapsids, which existed about 300 million years ago, they went on to evolve into mammals. Now, there were a multitude of changes involved in this process. One of those changes involved the development of the two remaining inner ear bones that are found in mammals, the incus and the stapes. Sorry, not the stapes, the incus and the malleus. So like the stapes, I should say, these two bones were derived from the bones of the jaw. Okay, so how did this happen? Well, in amphibians, reptiles, and birds, you have the quadruped bone, which is located here, and the articular bone. Those two bones connect together and are responsible for articulating the upper and lower jaws. So they serve as the site of articulation. In mammals, this articulation actually occurs at a different region. It actually occurs between the squamosal and dentary bones. So because it occurs at a different location, it essentially, quote unquote, frees up the articular and quadruped bones, which allow them to acquire new functions. So those structures are the remaining inner ear bones. The quadruped bone of the reptilian jaw evolved into the mammalian incus bone of the middle ear. The articular bone evolved into our malleus, and then the reptilian stapes kind of remained stapes and is the same stapes in mammalian ears. So the incus is homologous to the quadruped bone. The malleus is homologous to the articular bone, and the stapes is homologous to the stapes of the reptilian stapes, which is homologous to the hyomandibular bone. So essentially, the middle ear bones of the mammal are homologous to the posterior lower jaw of the reptile and to the gill arches of these early jawless fish. Pretty cool, right? Just some fun facts that you can throw next time you're at Thanksgiving with the family. Alrighty, so moving on to away from the comparative anatomy and embryology into next kind of area of evidence, which is observable evidence. So in some instances, we can actually observe evolution occurring around us. We can actually see that evidence. Some of them are these observable events that have been studied or documented. Others, again, are some that we're seeing occur in real time. And it shows that evolution is an ongoing process. Now, there are two kind of classic examples that are studied or talked about regarding observable evidence. The first was a study done by Peter and Rosemary Grant with medium ground finches. So they studied these finches for more than 20 years on the island of Daphne Major in the Galapagos Island. And they found through that 20-year period that beak depth variation actually occurred, and members of the population would go through these shifts in beak size in predictable manners, and those variations were correlated to different environmental conditions. So the actual beak of the medium ground finch, it's really short and blunt, and it's adapted to pick up seeds from the ground. Now, in 1976, the seeds on the island were very diverse and plentiful. And then, boom, in 1977, you had a drought, and seeds became much more scarce. Once all the small, medium-sized seeds had been eaten, the only ones that were left were these larger, spiny seeds that were very hard to crack open. And what the grants actually noticed was that the average beak depth started to change from one year to the next, again, in a predictable fashion. So during these dry years, or years where there were droughts, birds with deeper, more powerful beaks survived better. And then during the years with normal rains or normal average rainfall, the average beak depth decreased back to its original size. So this here actually shows the distribution of beak depths for those finch populations kind of before and after the drought. So having a large beak is most beneficial to the birds during the time of drought. And whereas having that short, blunt beak is beneficial for normal rain periods when you have a plethora of these small, medium-sized seeds. So again, these were dramatic changes in beak size, and they happened very quickly over time, which is very different from the slow, gradual evolution that Darwin had spoken of. The other prime example regards peppered moths and this concept of industrial melanism. So industrial melanism is a phenomenon that is known to have affected at least and well over 70 species of moths in England, but it's been best studied in these guys, the peppered moths. So prior to the 1800s, the typical moth of this species had a light speckled pattern, was this kind of phenotype here. The dark or melanic moth, or melanic morph as it's called, they were rare and were considered collector's items. Now, during the Industrial Revolution, soot and other industrial waste darkened the tree trunks, and they started killing off lichen. Lichen is the kind of precursor to moss, and it's that white kind of spongy looking material that grows on the outside of tree bark. So what people started to notice was that this light colored morph of the moth, again, the light colored morph is this guy. This is dark morph, light morph. So the light morph started to become more and more rare, but the dark morph, which originally was rare, was starting to become more and more abundant. To kind of give you an idea of how rapid this was occurring, I think it was in 1818 or 1819, the first melanic morph was observed and documented. By 1886, it was considered abundant, and your light morph, which had been considered abundant, was now considered rare. So just within that time period, you suddenly had that shift. Eventually, these light morphs, they were only common in a few regions, but those regions were far outside the zones of industrial areas. And it was thought that kind of the mechanism for this change, or the cause of this change, was selective predation by birds, where the birds kind of, they selected against camouflage coloration. So the moths that were not well camouflaged were those that were most predated. In the 1950s, a biologist by the name of Kettlewell did a release recapture experiment, or a series of these experiments, using both morphs. So he used both the light and the melanic morphs. And by observing bird predation from closed blinds, or these little kind of hidden tents, he was able to confirm that the conspicuousness of a moth greatly influenced whether or not it would be eaten. So the probability of a moth being eaten was directly correlated to how poorly it was camouflaged, right? So color success varied depending on kind of the wood type available. So in non-industrial woods, light moths did better. So here's your non-industrial woods. The tree trunks were light in color, they were not covered in soot, so your light morphs were well camouflaged, which means these had higher rates of predation. So in your non-industrialized woods, the light morphs were more inconspicuous, meaning they were less predated. Whereas in the industrialized woods, where the tree trunks were covered in soot and therefore much darker in coloration, the dark morphs, or your melanic morphs, were better camouflaged as opposed to the light morphs. So the light morphs faced higher predation rates than these guys, and therefore had lower survivability. So that's also an example of kind of directional selection as well, where kind of one phenotype is favored over another based on the environmental condition, and then kind of the frequency of phenotypes in a population will move in a trend towards whatever is favored by kind of environmental selection. All right, so last line of evidence is regarding biogeography. So again, biogeography refers to the study of where organisms live now and where they and their ancestors lived in the past. The geographic distribution of organisms, they follow patterns that are best explained by evolution, especially when they are combined with kind of looking at the movement of tectonic plates over geological time. So I like to say that biogeography is kind of this intersection between evolution and geology. So kind of for example, a lot of the species that kind of follow these worldwide distributions or global distributions, they're these broad groupings of organisms that had already evolved before the breakup of the supercontinent Pangea, which occurred about 200,000 million years ago. A great example of this is with the camelid family, or your camels. So they originated kind of in what is now the Pacific Northwest region of the United States about 40 million years ago during the Pleistocene. And from there, while you still had Pangea, or while the continents were still kind of all locked in formation of this one major supercontinent, they moved and kind of spread out geographically. There was still the land bridge here at the Bering Strait, so that's how they were able to cross into different regions of kind of Southeast Asia, India, and Northern Africa. And they also radiated out into the South Americas. Once you lost the land bridge here and these continents kind of separated from one another, these species were locked, or different species were locked onto different continents, and therefore kind of went through selective radiation events, which is why they all look very different from one another now. So in South America, you have four kind of different species of llamas. You have llamas, alpaca, vicuna, and guanaco. In North Africa, you have your Arabian camel. You can see all these different adaptations in these species based on the areas or the geographical areas they inhabit. But the reason why the camelid family has a global distribution was because they originated and they radiated out when these continents still formed Pangea. On the contrast, you can see other groups or other organisms that kind of are involved in these broad groupings that evolved after the breakup. And they tend to appear as uniquely or in uniquely similar smaller regions of the Earth. So, for example, there's unique groups of plants and animals that are split to either northern or southern continents, and they can be traced to the split of Pangea into two supercontinents, Laurasia in the north and Gondwana in the south. So you see this with a lot of plant species. So the Nothofaga species is principally Gondwanan in origin. We say that because of where it's located. So it's primarily constricted to the southern hemisphere, so southern lying areas in Tasmania, New Zealand, and kind of the southwestern coast of South America. And again, that the reason for such is because they evolved on Gondwana following the split of Pangea. So if they had arose during Pangea, there may be more of a global distribution, but they are isolated solely to Gondwana. Similar is with this type of flower. So you see it really well distributed, well represented in the southern hemisphere. So species that are kind of occupying below kind of this mark that I'm laying here usually represent Gondwanan evolution or Gondwanan origin. If you see them above or in the northern hemisphere, like you see with a lot of your bear species, that's more of a Laurasian descent or representative of Laurasian origin. So biogeography is also unique for looking at the evolution of species on islands, right? So that's kind of another great example. The big one being the adaptive radiation of marsupials in Australia. So placental mammals arose after Australia separated from the other continents. So only marsupials diversified on Australia or in Australia. Thus, most of the mammal species in Australia are your marsupials, meaning they carry young in pouches, while most of the mammal species seen elsewhere in the world are placental animals, meaning they nourish their young through the placenta. All right, so we will stop there. That is the end of part 2 and the closure of that whole lecture series on the evidence of evolution. And I apologize for the earlier distractions with Figaro and his squeaky toys. But I hope you found this lecture beneficial, and let me know if you have any questions.